Well, good afternoon, everyone. I'm Kevin Wu, Director of Product Management at Aldevron. Today, I'll be sharing how we're advancing mRNA vaccine and therapeutics through new innovations that address three of the biggest challenges in the field of mRNA manufacturing: scalability, speed, and cost. Aldevron's vision is a world where every cure is possible, and our mission is to partner with genomic medicine pioneers to revolutionize global health. We do this by offering high-quality products and services in plasmid DNA, mRNA, and proteins to the world's leading companies developing advanced therapies. Over the past 25 years, Aldevron has grown from a plasmid DNA manufacturer into a trusted partner across the genomic medicine landscape. We've manufactured tens of thousands of batches, supported over 1,000 advanced therapeutic companies, and contributed to the commercialization of 15 cell and gene therapies. We are particularly proud to have collaborated on Moderna's COVID-19 vaccine by supplying the linear DNA template. Our focus has always been the same: delivering high-quality, scalable solutions to accelerate life-changing therapies. Beyond custom manufacturing, we also offer a broad inventory of off-the-shelf products to help partners accelerate these therapies to the clinic. When we integrate our experience in custom services and off-the-shelf catalog, we can generate application-based workflows such as end-to-end mRNA drug product and RMP manufacturing. We reproduce material across three primary pillars, providing plasmid DNA, mRNA, and proteins supporting a wide array of therapeutic modalities and workflows. At the core of our business, we offer essential solutions and products, including analytical assay development and regulatory support throughout a partner's clinical journey. 3 years ago, we became part of the Danaher Life Sciences and Genomic Medicine Group. This growing network of companies share a unified goal: accelerating the advancement of life-changing therapies. Aldevron is proud to be an integral part of the synergistic network within Danaher. We are building value-driven work streams across Danaher Life Sciences companies. Together, we are at the forefront of advancing genomic medicines, encompassing sequence, generation, delivery, manufacturing, and analytics. Aldevron offers sequence-to-vial mRNA drug product manufacturing that simplifies the logistics and reduces risk. There is no need to coordinate shipments or timelines between multiple vendors, no multiple slot reservations to manage, and no inconsistencies in quality systems or release specifications. Everything is handled under one roof within a single quality system, ensuring streamlined execution and consistent quality product. With that foundation, let's talk about some of the pressing challenges in mRNA manufacturing. As the field grows, clients are commonly asking three questions: how can we scale? How can we go faster? And how can we reduce cost without sacrificing quality? I'll walk us through the advanced innovations offered by Aldevron to meet these exact needs. This starts with DNA and the plasmid template for RNA synthesis. We'll explore specific technologies that directly impacts scalability, speed, and costs. Starting with scalability, nanoplasmid is a next-generation plasmid vector with a small backbone, less than 500 base pairs. Larger sequences can be inserted without concern of compromising poly-A repeats. This increases plasmid yields, and nanoplasmids can be used as template for IVT without sacrificing on quality. And from a regulatory perspective, there is reduced risk because antibiotic selection is eliminated. A common problem using conventional plasmids in mRNA manufacturing is lower plasmid yields and loss of poly-A integrity, which affects downstream IVT quality and mRNA stability. We compared the yields and poly-A tail lengths using two plasmid vectors and compared traditional plasmids with retrofitted nanoplasmids. As you can see in the table, the nanoplasmid generated higher plasmid yields and also retained poly-A integrity. By increasing plasmid yield and preserving poly-A integrity, nanoplasmids can help ensure consistent and efficient IVT reactions. This improves the scalability of mRNA manufacturing. For plasmid to linear template, timelines often get bottlenecked. Our Alchemy platform takes a different approach. It's cell-free DNA, and unlike many other cell-free platforms, ours retains the original DNA template. We can directly use MCV, so there's no need to re-engineer and re-qualify starting materials. Most synthetic DNA platforms weren't originally designed with mRNA manufacturing in mind, which can lead to inefficiencies or additional processing steps. Alchemy was intentionally built for RNA workflows. Its cell-free and enzymatic system produces high-quality linear DNA template optimized for IVT. In the next few slides, I will walk through how this technology works, the key benefits it brings to RNA manufacturing, and how to integrate it with existing workflows to improve speed, flexibility, and consistency. Traditional E. coli-based workflows include cloning, screening, plasmid manufacturing, and enzymatic linearization, each step adding time. With Alchemy, we've condensed this workflow into a single enzymatic manufacturing step. Looking at the side-by-side comparison, traditional plasmids can take 3 weeks, 1 week each for material prep, plasmid generation, and linearization. With Alchemy, cell-free DNA is just 1 to 1.5 weeks, representing a 50% reduction in linear DNA manufacturing time. This can have big implications for rapid response programs and programs requiring iterative development cycles. It helps programs scale with more flexibility without compromising template quality. So what exactly are the features of Alchemy's cell-free DNA? It is a linear double-stranded DNA template that is open-ended, eliminating the need for additional capping or removal procedures. The cell-free linearized DNA sequences is identical to the plasmid DNA template, and this feature was specifically designed for easy transition from current plasmid-based processes. And like nanoplasmids, no antibiotic selection is required, providing an additional safety feature by limiting residual antibiotics. So how does this process work? We start with the plasmid DNA as a template and move into the enzymatic amplification process known as rolling circle amplification. Following the amplification, we use the same standard restriction endonucleases for the linearization. Following purification, the QC evaluation, the cell-free DNA template is ready for mRNA synthesis and IVT reactions. There's no additional optimization needed for mRNA production using Alchemy cell-free DNA when transitioning from traditional plasmid DNA linear template workflows. A common concern with cell-free linear DNA template is whether they can match the integrity and performance of traditional plasmid-based linear DNA. We collaborated with industry partners to compare Alchemy against standard pDNA-derived linear templates. As you can see on the left with template A and on the right with template B, the linear integrity is virtually identical between traditional plasmids and Alchemy-derived templates. And when used in IVT, the mRNA integrity is also comparable, or slightly better in some cases, with Alchemy. This confirms that Alchemy not only shortens timelines, but also maintains the quality expected for mRNA manufacturing. It gives mRNA developers confidence to adopt a faster, more flexible platform without sacrificing quality. The data shows that the same post-IVT purification used for traditional linear DNA works just as well for removing cell-free DNA. For both constructs, Alchemy cell-free DNA has significantly lower residual DNA levels than traditional plasmid DNA. For construct 1, 245 49 picograms of DNA per milligram of RNA versus 32 picograms with alchemy. And similar for construct 2, over 1,150 picograms versus 150 picograms of residual DNA for alchemy. One other advantage to point out here is the QC assays for the pDNA remain the same for alchemy cell-free DNA, since the DNA backbones remains unchanged in alchemy's process. This demonstrates alchemy not only accelerates template production, but also integrates efficiently into existing IVT and QC workflows, saving time. Alchemy is currently available as research grade quality and already in active development programs. We're on track for launching our GMP grade offering by the end of 2025 and will enable seamless progression from discovery to clinical phases. So keep an eye out. This is a platform designed for speed and scalability. So we covered scalability and speed with nanoplasmid and alchemy cell-free DNA. Both of these are powerful innovations in mRNA manufacturing. Next, I will talk about our high-cap T7 polymerase and how it cuts down on cost while maintaining high-quality mRNA production. HiCap is our optimized co-transcriptional capping mutant T7 RNA polymerase. This enzyme was engineered to produce synthetic RNA with improved safety and cost reduction. One of the major manufacturing challenges in mRNA therapies is unwanted double-stranded RNA during IVT. Double-stranded RNA can elicit an immune response and ultimately lead to cytotoxicity as well as inhibit protein translation. High-cap RNA polymerase significantly reduces double-stranded RNA formation compared to wild-type T7 polymerase, as shown in the figure by a lower double-stranded RNA signal intensity by dot plot assay. And this is across capping systems. This improvement in IVT purity produces a high-quality product that improves safety and efficacy. There's also potential for removing the polishing chromatography step, typically used for the removal of double-stranded RNA. This further reduces the cost for producing mRNA. Capping efficiency is critical in mRNA manufacturing as it directly impacts translation efficiency, mRNA stability, and immunogenicity. High-cap RNA polymerase was engineered to work with common co-transcriptional capping systems, significantly reducing the amount of capping reagent required. Comparing capping efficiency between high-cap and wild-type T7, we see comparable mRNA yields with greater than 95% capping efficiency, but requiring 62% less clean cap analog. For IVT reactions using ARCA, we see mRNA yields with a high capping efficiency, but requiring 20% less ARCA. High-cap RNA polymerase provides an efficient approach to mRNA production with significantly reduced co-transcriptional capping reagent, thereby reducing costs. In addition to improved capping efficiency and reduced double-stranded RNA byproducts, high-cap RNA polymerase also delivers consistently high yield and integrity across a broad range of transcript sizes from 2 to 9 kilobases. As you can see in the figure, the yields and integrity are equivalent or improved when using high-cap. This provides confidence across different mRNA therapeutic modalities without sacrificing quality, and we can even be used with mRNA-encoded therapeutic proteins. One of the main sources of immunogenicity in therapeutic mRNA is unmodified UTPs. To mitigate this, many developers substitute UTP with modified analogs like pseudo-UTP and N1-methylpseudo-UTP. As you see in the figure, high-cap RNA polymerase supports full substitutions with commonly used UTP analogs without compromising on mRNA yield, making it a strong option for reducing immunogenicity in mRNA therapeutics. So to summarize, high-cap RNA polymerases offers a strong balance of safety, cost efficiency, and manufacturing performance. It helps reduce double-stranded RNA byproducts, supports high co-transcriptional capping efficiency with less capping reagents, leading to significant cost savings, all while maintaining strong IVT and final product yields. HiCap is currently offered at research grade, and we're excited to be launching our GMP grade HiCap mid-2025. If you'd like to learn more, please come visit us at the Aldebaran booth. So we covered 3 key challenges today: scalability, speed, and cost, and the 3 advanced solutions offered by Aldebaran to address them. Nanoplasmids give higher yields without compromising sequence. Alchemy shortens timelines by up to 50%, again, without needing to compromise on sequence. HiCap reduces IVT costs by lowering capping reagents needed and improving quality and safety through reduction of double-stranded RNA. Together, these innovations enable smarter, faster, and more cost-effective mRNA manufacturing. Thank you for your time. Love to continue the conversation, and please come visit us at the booth, and happy to take any questions. And please come to the microphone if you can for the questions, so that our virtual audiences can also.
That was a great talk, Kevin. I'm from Codexis. I wonder, so when you compare the cell-free system with the cell-based system, for cell-free system, you use RCA. So traditionally, like, we can't control where it ends. So when you start to make long mRNA, so how do you control, there's no premature, like, for example, if I want to make longer mRNA, because your templates is, you don't know how short or long it is going to be. So is there any control on that? So that was my question.
Yeah, and thanks for that question, and it's probably better answered with our subject matter expert on that, but I will say that we are using the original plasmid templates in the RCA reaction there, and there is a cleavage site on there that as part of the reaction. So you're going to get this long, long DNA that gets cleaved afterwards for linear template.
What is the size you can synthesize with your platform?
The size of
the synthesizer DNA? What's the largest one you can synthesize?
This is for the cell-free DNA? Or which
for your
linear template?
Is that
Alchemy?
In the Reviver.
Yeah, so I don't believe there's any restriction in the size of the RNA
No, I mean
produce.
what's the largest? The total the DNA you can synthesize, you can produce.
In terms of this, I know for the scale of.
Is it up to 10 kb or shorter? Size of RNA, so the DNA, which size you can synthesize?
Yeah, again, I'm not sure about the exact size off the top of my head here, but it's something that I can refer back to our, again, subject matter expert to get the largest size for the linear DNA here.
So, okay. So in the other, you're using a Codex HiCap RNA polymerase.
Yes.
Same thing. Do you know what is the size for those RNA you showed?
Yeah, so what we're able to demonstrate on one of our slides there is we're able to manufacture from 2 to 9 kb without impacting, but I would say that that's not a limitation of what the length of the RNA is. It can be longer, but for our studies here, we were able to do from 2 to 9 kb.
Okay, so the other one you're showing, double-strand RNA, is reduced dramatically, the double-strand RNA with the dot block. So do you have a number? I couldn't see well, probably. Do you have a number? What's the level? It's a quantitation or?
Yeah, let me go back
relative,
to.
quantity. Yeah, this one.
not this one. Let me flip back to.
What is the point?
Yeah, we do have one of our subject matter experts in the crowd here, Kyle. Maybe you can comment on the level here.
It's on the low nanograms. Yeah.
What?
It's on low.
Nanogram per, alright. Nanogram per, per milligram, right?
Both the wild type entities or
It would be compatible. We don't see a difference whether you use the modified.
Can I? Yeah, nice talk actually. Your company is very outstanding in providing all kinds of, all stages of mRNA as we wish, you know. I'm very interested in your polymerase actually, that benefits the less production of double-strand RNA. So have you ever compared your company's polymerase with the thermostable T7 RNA polymerase in terms of double-strand RNA synthesis? The New England BioLab provides conventionally available the thermostable T7 RNA polymerase. And have you ever checked the?
Specifically double-stranded RNA?
Yes.
Yeah, compared to the thermostable from APP, they're significantly lower in double-stranded RNA.
So you didn't mention much detail about Codex high-cap RNA polymerase at this moment. You know, is this mutant RNA polymerase or?
It's a mutant
Okay,
T7.
so all right.
That's right.
Okay, so is it working in the ambient temperature, 37 degrees Celsius? All right. Okay. So you mentioned about the following step after the synthesis. If any necessary, you can purify double-strand RNA contaminant from your IVT.
That's
so
right.
what kind of method do you use in your company?
So we didn't talk about it on this slide, but we have cellulose-based.
Cellulose, yes,
Cellulose-based,
okay, yes,
and
so
then
just like
also-
BioNTech, okay, yeah.
We also have other chromatography-based methods for purification of double-strand RNA.
So what is the final outcome of double-strand RNA impurity, if any? Is there any
The cutoff there,
cutoff,
that's cutoff for double-strand RNA. It's a good question.
Yeah.
Typically, I would say that our clients are targeting between below 0.4 or 0.2% double-strand RNA.
0.2%, all right.
Obviously with therapeutics, mRNA therapeutics, it's lower the better.
Okay,
But I
yeah.
would say that 0.4 and 0.2%
Say
are.
0.4 is okay, I see, for your company, all right?
Yes.
Okay.
No question. So you use the arginine, that system?
Yes, the alchemy
grow
cell-free
in
DNA.
Reviver. Is this the data you come from? Is it from large scale or from the small scale?
No, so you're referring to the Reviver slide with with our nanoplasmid? I think this is the
So
your
this
question around.
data is from the large scale manufacturing, large scale, or from the small?
This is from small scale
small
manufacturing,
scale.
that's right.
Okay, thank you.
But we're not limited to small scale manufacturing. We're able to manufacture large scale and GMP as well.
Thank you.
And that goes for both, our alchemy cell-free DNA. Right now, it is in research grade, but I believe it's at 50 milligram scale at this point, but we're not restricted in the size of batch that we're able to make. And for GMP, I think we're targeting 100 milligrams up to 2 grams for the cell-free DNA.
Okay. So I'm interested in your Reviver cell strain. So are they, the growth, the median, everything is different from the regular median on the market?
So the Reviver is a proprietary cell line that we have, and it's been used with nanoplasmids with great success. Yes.
Thank you.
talk. My question is also on the dsRNA. So in order to measure the dsRNA, do you guys have any orthogonal method that you guys have confirmed this result with? Other than I believe this is
A dot plot method that we
Yeah,
have right
yeah,
now.
yeah.
Our analytical teams are working on other methods
Okay.
to measure double-strand RNA. We just don't have the data here to show at this point, but the work that we've done here is around the dot plot method.
okay, cool.
Yeah. Okay, well, thank you very much for your time.